The overall goal of this procedure is to transurethral induce mouse urinary tract infection. This is accomplished by first growing bacterial strains of interest and diluting cultures to the desired concentration. The second step of the procedure is to transurethral inoculate the mouse bladder with the bacterial strains of interest. The third step of the procedure is to collect urine samples and harvest kidneys and bladders. The final step of the procedure is inoculation of agar plates with urine and homogenized kidney and bladder samples. Ultimately, results can be obtained that show successful infection of the urinary tract based on the presence of bacterial colonies in plated samples.
Hi, I'm Michael Hsieh, and welcome to my lab in the Department of Urology at the Stanford University School of Medicine.
I am Thathireddy, also from the Hsieh lab.
Today we will show you a procedure for mouse transurethral catheterization.
We use this procedure in our lab to study the induction of urinary tract infection in mice.
So, let's get started.
In a laminar flow hood, cut a 30 centimeter length of polyethylene tubing. Secure a 30 gauge needle on a sterile 3 cubic centimeter syringe. Use sterile forceps to pick up one end of the cut tubing and slide it onto the needle until it meets the hub. Cut the tubing so that approximately 2 centimeters extend beyond the tip of the needle. This helps to prevent injury to mice during catheterization. Remove the catheter from the syringe and place it into a sterile petri dish. Make one catheter for every mouse to be infected. Sterilize the catheters with ultraviolet irradiation for at least 30 minutes. Seal the dish with parafilm to keep the catheter sterile for long-term storage.